You can hear some function of them. One of them is the cells or particle or microorganism that enter to the cell by endocytosis, digest them and remove them. They are important for the defense and also immunity. They are organelles that damage or no, they are the old organelles. These lysosomes can recycle the old or damaged organelles. And finally, the main function of them is the turnover and renew of the cellular component, different organelles or different structures that damaged or aged. That the particle or the damaged organelles fuse with lysosome after fusion by their enzyme, they lysis and digest it. Lysosome have membrane, and the enzyme of the lysosome for their function need the acidic pH. Around, the pH of the cells in the physiological is there between the 7, around the 7. But for the function of the lysosomes, the pH of the cell should be acidic between 3 to 4. Why is the endocytosis of particle or these damaged organelles? They expose some markers, some as proteins that act as an alarm and send message for the lysosomes that they should be removed by the cell and the lysosome digest them. As I explained for you about the name of the lysosomes, that the bacteria or microorganisms come, they named as endosome. The lysosomes then don't fuse for any particle or anything named as the early or primary lysosomes. After fusion, they named as secondary or late lysosomes. When they digest the cell, the damaged organelles or damaged component of the cells, because they digest itself, the particle that form by fusion of the lysosomes and other organelles named as autophagosome. It's a particle that formed by the fusion of the lysosome or some organelles or some component of the cells. Sometimes all of the cell damaged or the life of the cell ended. Yes? Now the lysosomes kill all of the cell. That is named autophagy or autosuicide. That the cell kills. The autophagy is one of the important subjects that today the scientists interested to eat, because they believe that the autophagy in our cell is good for us. It's necessary for us for the removing of the harmful cells or mainly in the tumoral cells, yes, in the cancer, that the cell in the first step or in the low grade, grade 1, 2 can removed by the cell. Protoazole is a type of a special lysosome. Don't have any membrane. As you see here, it has a cylindrical structure. The structures of these organelles as this, that we put 4 rings together. That these rings, the rings is another type of.
Excuse me. Please, we have lysosome already. What that's digested nutrients like proteins. So why do we need again something like proteasome?
The lysosome, yes, remove.
Lysosome.
Yes, yes, have protease and other type of enzyme. Remove the particles that enter to the cell and also some damaged protein. Yes, they are damaged or old. Yes, but proteasome remove the proteins that not damaged, but just after synthesis, some modification not occurred there. Yes, but mostly the lysosomes remove the damaged organelles and damaged structures totally. But the proteasome just remove the non-functional polypeptide, just act on its role in the removing of the peptide just. But the lysosome involved in the removing of other components, other macromolecules or damaged structures. Peroxisome is another specialized lysosome. It's a zone that involved in the removing of the peroxide hydrogen. Peroxisome. You know, the peroxide hydrogen is a biochemical material that normally broken to the water and oxygen. Oxygen in our body normally have two atoms. Yes? But why is the metabolysis of the H2O resulted to one oxygen? This oxygen is harmful. We name them as, maybe you also hear reactive oxygen spices. Or I don't know, maybe use here the ROS. Yes, these are some, yes, the oxygen that have just one atom, as these oxygen are harmful for the cells. The peroxisome function is the removing of these oxygen after the metabolization of the hydrogen peroxide that do this function by some enzyme that the main important of them is the catalase. I think you hear the antioxidant. Yes? Now today lots of scientists, scientific nutrients, yes, recommend to us that we eat antioxidants that are in the foods, in the fruit, in the vegetables, or no. Some money involved in the production of the antioxidants, such as, for example, the melatonin, the curcumin. Yes, there are the 10 of them in the pharmacy. We can buy them and eat. Antioxidants are some maybe natural or synthetic material that also involved in the removing of the oxygen in our body. Because of the today, recently, because of our lifestyle, our nutrient, our body have a lots of oxygen. And these hydrogen peroxide, these their peroxisome with its enzyme may be unable to remove all of them, and maybe we need to eat some supplementary tab or capsule or foods for removing of them. This function of the peroxisome is generally occurred in all of the cells, but in some cells they also have the specific function, especially in the liver or hepatocyte, liver cells, that they included some enzyme that involved in the lipid metabolism and also oxidation of the fatty acid, and also involved in the synthesis and production of bile in the hepatocyte. The another organelle in the cell are cytoskeleton. Skeletal, you know the skeletal system. Yes, included our bones that maintain our shape and also help to us for different functions. These cytoskeleton also included a lot of the shape of the cells. Or in all of the cells. Microtubules have more diameter. They included the tubulin proteins and involved in the intracellular transportation, involved in the movement of different things from one organelle to another. Intermediate filament diameter is between of them. These filaments are not in all of the cells. They are just in some cells. The monofluorescent that they stay by the fluorescent dye, and here you can see the actin filament, as you see, are under the cell membrane. The microvilli structures keep the microvilli structures and also the shape. Microtubules are in the cell involved in the, for example, transportation of the vesicle to other one. An intermediate filament that in each they are just in specific cells that I explain for you later. Microfilament are thinnest part of the cytoskeleton, included the actin filament. That is, actin filament form a double chain of the actin globular. Yes, the actin in the cytoplasm are as some small bars, yes, that are spayed. When they want to attend and do its function, they attach together and form a fibrous chain of the actin filament. In most cells, this silent form. They form a thin sheet or network under the cell membrane, keep the shape of the cell. In skeletal muscle, they integrated to myosin involved in the muscle contraction. Also, these microfilaments can involve in the cytokinesis, in the division, mitosis division, when occur one cell divided to two cells. That for the separation of two cells, again, this filament attend. Microtubules. Their subunit is the tubulin molecules, that two of them join together and form a heterodimer of tubulin. And finally, they polymerized and arrange a line together and form the microtubules. Microtubules in our body, the first function of this is the intracellular transportation. As one example, for the transport of some things in the city or between the city, yes, there are some roads, yes, that the transportation of things, person, people, or others done by some cars. This is cars, this is the road or a street, yes. Car for their movement need to get oil or gases, yes, that these oil or gases act as a motor, movement, and finally move the car, yes. This example is about the function of the microtubules. Microtubules are roads of the cells, are in different parts of the cells and move there, for example, vesicles from RER to Golgi, from RER to mitochondria. Golgi apparatus package the mitochondrial proteins, that these proteins should move from the Golgi apparatus to mitochondria. The microtubules act as a road. The vesicles are car and for the movement and act as for the motor and movement of them. There are some motor proteins that join to the vesicles and move them. The motor proteins have two types, as kinesin and dynein, that carry out the vesicles along the microtubules. The microtubules, vesicles, and dynein or kinesin. The kinesin some inner, the ATP synthase or other things that produced in the other organelles, and they should conduct them in opposite direction or in the as retrograde direction. Yes, for this the dynein carry basically in the opposite direction of the kinase. The main function of the microtubule is the movement of the different things. For example, oxoplasmic transportation that we know about more in the nervous tissue. Melanin transportation from melanocyte to other cells. For the movement of the chromosome in the mitotic division and basically movement in different cells. Also, microtubule attend in the formation of other organelles in our body, such as cilia, flagella, centriole, and basal body. The cilia, flagella, centriole, basal body, all of them included the microtubule. It means microtubules involved in the formation and function of these organelles. You have them, I think. Centriole involved in cell division, yes? They included the microtubules. The microtubules are as tubules. 3 of them assemble together and form a triple structure. 9 of these triple structures, 9 of triple microtubules form the centriole as this. That they again are paired and located vertical together and involved in the cell division for the formation of the division spindle and finally separation of the cells. Cilia and flagella. Do you know them? Cilia are some microtubular structures that are in the apical surface of some cells. Move as a
Like the paramecium.
Huh?
Like the paramecium in the sea.
Yes, and move the materials, move the mucus, move the particles. There are a lot of them in the respiratory system, in the nose, in the trachea, with the breathing, yes. We breathe a lot of harmful particles, dust or others. These cilia move and again out them from our body. Flagella in the human is just in the spermatozoa. Male gamete move the sperm from the male to female. The structure of the cilia and flagella also included the microtubules. Here, 2 microtubules assembled together. 9 of double of them, plus 2 for the cilia and flagella. Transversation of them under electron microscope. In the centriole, 1, 2, 3 microtubules. 1, 2, 3. 9 of them, yes, assembled around together and form the centriole. In the flagella and cilia, 9 double of them in the peripheral part and 2 single of them in the central part. 9 double plus 2. Arrangement of the microtubules in the cilia and flagella. In the cilia and flagella, one of these microtubules also attached to the dynein. Dynein is a motor protein that move the flagella and cilia. If the dynein damaged or mutant, some genetic disorders, the flagella and cilia biotin, desmin, perial fibrillary acidic protein, neurofilament, and lamin. They are just in some proteins, not all, except lamin. Lamins, again, are under the nuclear membrane. In the cell, have nucleus, as same as two actin filaments under the nuclear membrane, included the lamins that keep the shape of the nucleus. But other one of them are just in one type of the cell. Cytokeratin or keratin are just in epithelial tissue. Not, for example, in the nervous tissue or in the muscle tissue. Biotin and desmin are in the smooth muscle cells. GFAP, and neurofilaments are in the nervous tissue. These cells, as you see, have just one of them. The intermediate filaments are important for the distinguish of the origin of the tumors. For example, one person suffered from sarcoma. Sarcoma means the tumor of the muscle, yes, muscle tissue. But the physician want to know that these tumor originate from the muscle or no, metastasis from the other tissue. They do some staining, for example, for the cytokeratin. If they find the keratin, it means that these tumors originate from epithelial tissue and metastasis to the muscle tissue. Clinically, these intermediate filaments are important. Additional to organelles in the cytoplasm are some inclusion that they are non-motile and that are no metabolic activity, included fat droplets that store the fat or lipid, glycogen organelles that accumulate the carbohydrate, are more in the muscle tissue cells and also in the hepatocyte cells, and some pigmented granules such as melanin, eumelanin, lipofuscin, or others that give some color to some part or some cells. These granules or droplets, yes, don't have main function, special function, as same as two organelles, but they are important and necessary for the function of some cells. For example, melanin in the skin, in the hair, in the nail, yes, are important, necessary for the protection of our body. Or in the retina, they are necessary for the vision. But they are allowed, they don't have any function allowed. They need to another organelle or another thing. Just some minute. Rest, if you have question, ask me that. The last one is there, we close. My speaking is okay for you? You can understand the topics?
We are just so
What
fast
else?
actually.
What?
We are just so fast, you know. Today
I'm sorry for this, because this is your schedule.
yes.
Because the students that are here, we have this problem because we come from Tehran and your classes is very compact, and also about you because your previous time, your class cancel, and you must have a lot of class from today until summer. Just I want to change that, put them in the week, but Mr. Darabi keep the Friday that you have a little rest for these topics. Yes, I put in the desktop. If when I come back to Tehran, if I access to the NANIT system, also I uploaded the PDF plus my voices, because I have the voices in Tehran.
How come you get it?
may be changed. That we know about that tomorrow in case superiority. Nucleus included the DNA or chromosome that you know why the DNA and chromosome. Nucleus control all of the function of the cells, and because all of the cells have nucleus, we can tell that the nucleus control all of our body function. It have different part as membrane, nuclear membrane, that also have another name as nuclear envelope. Mostly we use the nuclear envelope inside of the nuclear membrane. Inside of the nucleus there is the nucleus and also the chromatin, that is the heretical molecule in the body. Nuclear envelope consists of two concentric membranes. The structure of the membrane is similar to the cell membrane. Again, phospholipid bilayer and the proteins. The outer nuclear membrane continues with the RER that I said for you in the endoplasmic reticulum. And the inner nuclear membrane associated with lamins, intermediate filaments that keep the shape of the nucleus and also involved in the formation of the nuclear pores. Nuclear pores are some small pores with very narrow channel that involved in the nucleocytoplasmic transportation. Transport something from nucleus to cytoplasm, such as ribosome, such as RNA, and in the other hand, the proteins that involved for the synthesis of the internuclear cells. They form included 8 proteins that involve just in the trafficking of the nucleocytoplasmic trafficking, that the lamin filament are necessary for the formation of chromatin are inside of the nucleus. There are some chromosomes in the human that included the DNA and some proteins. The chain of the DNA and some proteins that join to them. Chromatin, that is the subunit of the chromosomes, based on their attendance in the transcription or no transcription, divided to heterochromatin and euchromatin. Chromatin are chainy structures that coil together. I explained at first this one, then come back to previous slide. The smallest subunit of the chromosome are nucleosome. What is the nucleosome? One chain of the DNA plus some proteins. What is the function of these proteins? The DNA have chainy structures. If in our body the DNA remain as chain, yes, they are don't have space for their location. We can cover all of the Earth by 1% DNA when they have the chain structures. For compaction of them, these chain structures change to the circular structures by some proteins that named histone. Histones are structural proteins that change their chain structures of DNA to circular or coiled structures. That 4 pair of them are inside of the structures of the nucleosome. That they are 4 histone in 4 pair as H2A, H2B, H3, and H4. And H1 link the origin, the start and end part of the coiling, the euchromatin written open, not very coiled. Why? The euchromatin involved in the transcription is active pattern of the chromatin. But heterochromatin is passive or inactive or rest form of the chromatin, not involved in the transcription. These two can change together. We have in all of the cells heterochromatin and euchromatin. Euchromatin show the activity of the cell. Every cell have more activity, more euchromatin, such as neurons. But some of them are not very active. For example, the digestive cells. Yes, when we sleep. Yes, they are work, but not very active or not very fast. Yes, in this cell, the pattern of the heterochromatin is more than the euchromatin. And finally, the chromatin again compact and coil more and more and form the chromatin, and finally they form the chromosome that we can see under microscope. The chromosome with special staining, hematoxylin and those. Thus, the heterochromatin and euchromatin are two different patterns of chromatin based on its activity. Heterochromatin mostly located in the peripheral part of the nucleus. They are more coiled, and because they coiled and form a glomerulus structures, then they don't allow, they don't any permission to the transcription. But when they open, as euchromatin, the transcription can occur in the euchromatin. Under electron microscope, heterochromatin, electron dense, euchromatin clear. With H&E staining, heterochromatin have purple color, dark purple color, and euchromatin is clear. The euchromatin show the activity, high activity of the cell. Nucleus is a spherical and highly basophil structure in the nucleus that, as now and do now, the RNA and also ribosomal subunit synthesis done in the nucleus. Under electron microscope, this picture shows the neuron, one nerve cell with euchromatin nucleus. Euchromatin nucleus means all of the chromatin are you. All of them are involved in the transcription. And because this nucleus is clear, we can recognize the nucleus in this cell better. You know the cell division? Yes? If you don't know, I'm sorry, you should study by yourself because they, and what I see you now, there are two types of them, mitosis, meiosis, or what. But for the cell division, for the formation of two cells, the stem cell are some basal cell that can do mitosis, replicate themselves, and also some of them differentiate to other cells. For the mitosis, or the cell that want to any question, can ask me. Because if you look at more details, I don't necessary for you. And this is the briefly of the neutrons and
Öyle inanıyorum.
check your area. Endoplasmic reticulum that included some cisterna that going together that we want to know about them. Ribosomes have two subunits that, as I said, when they are not involved in the protein synthesis, these two subunits are separate from together. When they attend in the synthesis, these two subunits attach together the cell signaling from very simple. But how cells communicate together? As I said for you and you now, in the cell communication, there are at least two cells. In the endocrine signaling pathway, the signaling cell and target cell are far from together. Yes, we have this signaling pathway in the endocrine system, in the endocrine glands. Pituitary gland release growth hormone. Where are the growth hormone receptors? In the muscles, in the bones, yes. They are far from the pituitary glands. This pathway named as endocrine pathway. Endocrine cells release the signaling molecule as hormone to blood stream. And the receptors are in target cells that are far from them. The next one, paracrine. Schematically shown here. Anybody can explain about it? About the signaling cell and target cell? They are neither together. They are located in one organs. Yes? Signaling cell, signaling molecule, the target cell located close to it, such as G cell in the stomach that produce one hormones as gastrin that have receptors on the other cells in the stomach. This pathway named as the paracrine signaling. They are located in the same locus. That is, the paracrine is important for the embryonic development. Juxtacrine. They are located adjust together. They have physical contact. The signaling cells, signaling molecule, the adjust cell that attach to the cell have receptor and act as a target cells. They have direct physical contact in the juxtacrine pathway. The other type of signaling pathway is autocrine. It means auto, itself. The signaling molecules have receptors for the signaling molecules. Maybe they are same cell or similar cells. Yes? Another cell adjust to it. This is not paracrine because they are similar cells, same cell. In the autocrine, the signaling cell and target cell are same. Insulin growth factor is a factor that acts as the same as the growth factor, growth hormone. Yes? Insulin growth factor acts its function as this, as autocrine. And the final pathway. That about this pathway there is a challenge. Some of the scientists believe that is a type of signaling pathway, but because just occurred in the nervous system, some scientists believe this is the function of the neurons, not the signaling pathway. But here, in this pathway, there is a nerve cell or neuron that releases its product as neurotransmitter and conduct it into other cell to target cell. This target cell may be another nerve cell or the muscle cell or other. In this pathway also, the neurotransmitters that act as a signaling molecules act on the adjacent cells. Maybe because some of them believe that is a type of Hughes-Lateran pathway. Not neurons. The ligand, different type of the ligands that act as a signaling molecules have receptors to other cells. The type of the receptors, some of them are on the cell surface and some of them are inside of the cytoplasm. There are the two types of receptors, cell membrane receptors or intracellular receptors. There are receptors that are on cell membrane, just I show to you because in the physiology explained for you that this picture shows that when the ligand join to its receptors, how act on receptors, how act on the cell. Maybe open the ionic channel, maybe open the ionic channel or activate the enzyme or activate a G protein. G protein is a protein that dephosphorylated GTP, guanosine periphosphate, to GDP. Yes, 1P is one type of receptors are the cell membrane that is very usual in all of the cells that are different type of the receptors on the cell membrane that you know in more detail in the physiology. But some of the receptors located inside of the cell are intracellular receptors. For which type of the signaling molecules? The signaling molecules can pass easily from the cell membrane without using of the energy. Which type? The steroid hormones. Or the material that biochemically are lipid. Because the cell membrane structures included the phospholipid, they can dissolve in the cell membrane, pass from the cell membrane by diffusion. They don't need to any receptor on the cell membrane, but they receptors located inside of the cell cytoplasm, such as estrogen, testosterone, the adrenal gland hormones, that they have intracellular receptors in the inside of the cell, inside of the cytoplasm. They attach to the receptors and activate some cascade inside of the cell. Any question? We have a rest until 11, yes, 15 minutes. Then I continue for cytoplasm. I hope you don't confuse and you don't tire for the session.
Until 3 p.m. we have our lecture, right? Until 3 p.m.
No.
I don't know because maybe around if we continue following together, if you want to eat lunch and have a break between 12 or 1, yes, I think around 2 is finished. But if we continue following, we finish I think in 1. But I
Ahmed, we have Persian class
some
today.
information. Yes.
Do you have a Persian class
Yes.
today?
At 3:30.
Yes, we finished at 1 as well.
That's okay, there are two.
لا الزلمة
We have a
Persian class.
ساعة
11
Yes, because in your schedule you have the class until 3 o'clock, but I think it's finished at 2.
Right.
هي
And
تسجل
after the break, yes, I explained about the circumference, but I see not finished under 12. We have a class until 12, and then we have a rest time for the lunch, break or other, and we start again in 1.
ساعة بريك
Pass from the nuclear core, come to the cytoplasm and translate on the RER or in the right. That is included some the amino acid, the other amino acid also come by transfer RNA, they switch together and finally form a peptide chain. But one thing that is important, the proteins that are necessary for the synthesis of ribosomes or mRNA synthesis again in the ribosome, in the ribosome, then again pass from the nuclear core go to the nucleus. Yes, the proteins. Yes, different type of the proteins that needed for the replication, needed for the synthesis of the ribosomal subunit or other, come from the cytoplasm to nucleus and nucleolus, and then different type of RNA and also ribosomal subunits synthesis in the nucleus and nucleolus, and then they come back to the ribosome. There is a two-way for this factor. Ribosomes have two types, as I said for you. Some of them are free, that join together by mRNA. Very polyribosomes synthesis proteins remain in the cytoplasm. For example, the proteins, the enzyme of the lysosomes. Some of the proteins that are in other organelles, in the mitochondria, Golgi apparatus or other, synthesis in the free ribosomes. Some of the ribosomes attach to the ER, endoplasmic reticulum, is another type of the organelle. They are bound polyribosomes. They synthesis the proteins that segregated in the ER and exit from the cell. Maybe exocytosis and used by another type of the cell. Most of them are cell membrane proteins, all of the cell membrane protein synthesis by the ER. ER is a network of tubular or circular structures in the cell. That this network included some tubules, yes, maybe some of them have narrow lumen. They form a tubular structures, but in some of them, these lumen a little expanded and formed a circular structure. They are the sac-like structures. They connect together by very narrow and small canals. All of these structures named as cisterna. Cisterna means some channels or sacs that continues together and connected together by small canals. The ER based of the existence of the ribosome or absence of the ribosome divided in two types. Rough endoplasmic reticulum have ribosome, expanded and have a circular shape. They are close to the nucleus. Why? Because the ribosome synthesis in the nucleus pass from the nuclear core and join to the ER. Also, messenger RNA, RNA, or other type of the RNA. In the human body, in all parts, the human body have an economy for itself. Do all of the function with low energy, with low using energy. For the here, yes, the low distance between the nucleus and ER, yes, the ER use low energy, low ATP for its synthesis. But we want to know how mRNA, yes, mRNA in the nucleus, yes, come to the cytoplasm and have two-way for synthesis in the ribosome, yes, and RER. How mRNA understand that should go to RER or to free ribosome? The mRNA that involved in the synthesis of the protein in RER have a small peptide that named as signal peptide when exit from the nucleus. Yes, the mRNA, the free ribosomes get attached to this mRNA, join to subunit of the ribosomes together and start to synthesis of these small proteins that named signal peptide. Signal peptide act as an alarm for a particle that recognize this peptide. There is this particle are in the cytoplasm as signal recognition particle. The particle that recognize this signal peptide. The mRNA that included this signal peptide recognized by this particle that in abbreviation named as SRP. Join to signal peptide. After joining of the SRP to He said that the RER synthesis, that proteins that not remain in the cell. About the lysosome enzyme, they have the exception. Yes, the lysosome enzyme. Or the granules of leukocyte. The basophil, neutrophil, eosinophil are different type of white BC, white blood cells, that included a lots of cytoplasmic granules. These granules synthesis in RER and remain inside of the cell. Maybe this storage is temporary. They synthesis and store in the cytoplasm for several hours, several days, and then exit from the cell, such as pancreatic enzyme. The pancreas is a gland in the GI system, synthesis some enzyme that when food received to the second part of the duodenum, yes, send impulses to the pancreas and the enzyme released from the cell by exocytosis. This is the temporary reservation. And the integral membrane proteins. The proteins of the cell membrane, all of them synthesis in the RER. SER, smooth endoplasmic reticulum, no ribosome. The cisterna have tubular structures. Their lumen is narrow, and they also connected together by very small channels in various shape and size. What is the function of SER? Generally, the SER synthesis the phospholipids of the cell membrane and other membranous organelles. Generally, the lipid synthesis in all of the cell done in the smooth endoplasmic reticulum. But the SER in some cells have another specific function, such as in the liver cell, hepatocyte, involved in the detoxification. They remove the poisons from the food, from the drugs or others in the hepatocyte. Store and release calcium ion in the skeletal muscle cells. Additional to lipid production, they also involved in the storage of the calcium ion in muscle cell, not other cells. That these phospholipids that synthesis in the SER transfer to the cell membrane or other organelles, maybe by vesicles, yes, some vesicles separate from the RER or SER and join to the Golgi apparatus or go to other organelles, or some of them have a carrier. That these carrier are some proteins that carry the phospholipid to the cell membrane. This is two-way for the transportation of the phospholipid. But the proteins that synthesis in RER or ribosomes, all of them transfer by vesicular transportation in the outside of the cell or in the inside of the cells. The another one of the organelles are Golgi apparatus or Golgi organelle. The Golgi included some cisterna that generally are close to the RER and SER. Again, this reason is because of the using low energy for transferring of the vesicle from RER to Golgi apparatus. Another name of the Golgi apparatus is the sheepan. That have the butter, cream, yogurt, or others. Yes? Then these products packaged in different shape of the package. Some of them are small, some of them are big, some of them are in the paper pocket, some of them are in the metal pocket, yes? Packaged in the factory. And then sent to different part of the city. Or no, maybe sent to different city. For example, in Iran, the Pega factory is in the Gilan. Different product of this factory sent to Tehran, Mashhad, Isfahan, or other thing. Yes? In the Golgi apparatus, occur this example. The proteins enter to the Golgi apparatus. The cisterna that is close to the ER, here is the ER. Yes? The first cisterna recept the proteins, accept the proteins, named as cis face or cisterna face, that located close to the RER and the vesicles that coming from the ER or RER or SER attached to the Golgi apparatus. Yes, as some proteins or lipids. The post-translational modification occur to the different cisterna of Golgi apparatus, as assembling, breaking, and glycosylation, sulfatation, I don't know, the phosphatation, different adding of the glucose, phosphate, sulfate, or others. All of them occurred in the Golgi apparatus. Finally, they packaged as some secretory vesicles in transphase or transverse phase. The secretory vesicles that here detached from the Golgi apparatus included some modified proteins that should carry out to the other part of the cell, yes, or exit from the cell, yes. The packaging of the proteins done in the Golgi apparatus. This secretory vesicle addressed one of them, for example, the exon should to go the lysosome, included the lysosomal enzyme. One of them included the mitochondrial protein, should go to the mitochondria. Addressed to them. All of these are the function of the Golgi apparatus. For transferring of the materials from different cisterna, because this cisterna not connected together as cisterna of the ER. Yes, in the ER they interconnected together by very narrow and small channel. But the Golgi apparatus cisterna as some pancake located under together. Yes, they don't have any interconnection. For the transferring the vesicle from, for example, this cisterna, attach to this one. And again, detach and attach to another one. The transferring of the vesicle in the Golgi apparatus done by attachment and detachment of the vesicles. They are not interconnected together. But finally, these vesicles, as secretory vesicles, detach from Golgi apparatus. Maybe they exit from the cell by exocytosis, such as in the mast cell, in the eosinophil, in the white blood cells. They exit from the cell and do their function in the ECM and also connective tissue. Some of them from Golgi apparatus or no, some endocytotic vesicles enter to the cell and fuse with their lysosome, and lysosome digests them. All of these transportation done by vesicles. The vesicles are one type of organelles that enclosed by membrane and transfer inside of the cell by motor proteins and also the microtubules that explain for you later. The vesicles that enter to the cytoplasm also, when they are not fused by two lysosomes, also named as primary or early endosome. Endo means in transome. Is a postfix word that used for a small particle in the cell as endosome, ribosome, lysosome, peroxisome. Yes, we know about them in later slide. All of these are a small particle or a small organelles that generally named by zone. Lysosome, ribosome. Lysosome means the organelles involved in the lysis. Ribosome, ribonucleic acid zone. That because this particle, this vesicle enter to the cell, also named as endosome. Before joining or fusion to the lysosome, any changes occur then, named as the primary or late and early endosome. After fusion with lysosome, they also named as late endosome or secondary endosome. They have equal and also same function but have different name. For example, one person is and I don't know his name is Hussein, but in the family call Hassan. His friends call him as the Ahmad. I don't know. Yes, have a lots of name, but it's a one person. Yes, here also as this. Yes, the early endosome is a endosome that just enter to the cell, not fuse, not anything, but have different name in different books. Please, I want to know. Maybe you, for example, search in the internet and see another name. They are equal. Vesicle. Now then, yes, are small enclosed membranous particles that synthesis in the ER, in the Golgi apparatus, maybe they enter by the endocytosis as secretory vesicle, transporting vesicle. All of the membranous small organelles are vesicle. Mitochondria. I'll explain this one and then we have the rest. Mitochondria is a double membranous organelle. Exception of the mitochondria have two membranes. Generally have oval shape in all of the cells, except in the adrenal glands they have round cells because the mitochondria are powerhouse factory of the cell. They involve in the production of the ATP and energy. But in the adrenal they have another function additional to the ATP synthesis that you know, inshallah, in the next semester, the third semester. Mitochondria have a double membrane. These two membranes separate from together by very narrow space that named as intermembrane space, located between of the inner and outer membrane. In the inside of these proteins involved in the ATP synthesis. But when they pass from the thermogeny, they involve in the heat production. The mitochondria is a one organelle that involved in the regulation of body temperature. These proteins are more in the mitochondria of the hibernate animals, that they sleep in the winter for a long time. For the regulation and control of their body temperature, this protein is very important. In the inter or inside of the inner membrane, this space named as the mitochondrial matrix. That this matrix have a jelly-like structures, contain a few amount of the DNA, RNA, some enzyme, and also the water. But their mitochondria have another exception that can do mitosis, that can break and produce more mitochondria, and also can increase the number and length of the crystal when the cell need more and more energy. But this is important. The protein synthesis in the mitochondria occurs in the small, and the mitochondrial protein produced in the RER and transferred to the RER. For example, the membranous mitochondrial proteins, all of them synthesis in the periribosomes mostly, and also RER, and then transferred to the mitochondria. The RNA and DNA are very, very few and not enough